Hey, good morning, everybody. Can you hear me? In the back, can we hear me? All right, good morning, everybody. How are you today? Thank you all for coming out to the 2024 Research Partner Thank You Lunch. This is probably my personal favorite event of the year each year. We missed a couple of gaps in the COVID era, but it's a thrill to see this return as a true annual event. Just some programming notes and announcements first. I know most of you are used to our co-director, Dr. Jason Karlowicz, emceeing these events. Unfortunately, Dr. Karlowicz's father passed away yesterday. He asked that I share that news directly with all of you. He has spoken about his father many times in public settings and academic presentations, and he regrets that he couldn't be here, but he plans to continue speaking about his father's story and how it impacts the work of our center and the care programs that we all support. So with that, I also recognize that I am in the lucky position of being between the food that brought you in and the information that caused you to register in the first place. So I'll keep it very brief. But my name is Terrence Casey. Most of you refer to me as the one with all the emails. And I accept that. Yes, I'm our communications director, and I lead our outreach operation as well. And really thrilled to welcome you all here. We're going to have a kind of a different structure than you're used to if you've been here before. We'll actually lead off with our keynote address from our other co-director, Dr. David Wolk, in just a moment. Then we'll turn it over to some of our other leaders for updates on various components of the center. So the ABC D² study, the Aging Brain cohort dedicated to diversity and study. We won't be pleased on that. An update on clinical trials from Dr. Vaishnavi. An update on our social work and care programs, and then I'll come back and just give you some brief introduction to how you can keep in touch and some exciting developments in communications. Then, of course, we'll wrap up, as we always do, with the Q&A with our panel of speakers. So start thinking about your questions. Please do save them until the end. We will have 3 different microphones going around the room, so plenty of opportunity. Just a couple of quick reminders. At the registration desk, I know there was some backlog, so you may have missed some items. One, if you parked and you parked in the garage at the Sheraton, please make sure you go back and get validation so you'll get a discount on that parking. 2, at the other end of the table, some people may have missed it or not known what the heck it was because it was all folded up. But we got some Penn Memory Center branded cleaners here. These eyeglass cleaners, screen cleaners. Please feel free to snag one on your way out if you didn't already get one. Or if you did, we won't tell. But before we do anything else, I just wanted to make sure on behalf of the center that we all say thank you for being here and for your continued participation in our research. Our research participants and their care partners and their study partners make everything else that we do possible and worthwhile. So thank you so much for all of the effort that you put into the research. We recognize that a couple of sandwiches and chips is not exactly a fair trade, so you're still coming out as the true altruists here, and we truly value that. I'd also like to make sure that we're including in here our community partners and our community advisory board members. This event really is for the research partners, and I respect that, but our community advisory board members are members of our newly formed participant advisory board, the community partners in Philadelphia with whom we work to develop these different studies have made everything possible for you to come in to visit and have the optimal experience. So thank you to all of them who are here. And just as a last final thank you, just want to personally acknowledge the staff who made this possible today. I lost count, but we were up over a dozen at least, and in particular, Marie Engineering, who's our events assistant, and is probably busy doing something right now. She's in the back there. But Marie organized this whole event, worked with the Sheraton, communicated with all of you. So just a special round of applause for Marie, please. And with that, I'd like to welcome Dr. David Wolk, our co-director of the Center, for our keynote address.
Hello, everyone. Just to echo Terrence, we are incredibly grateful and thankful for you all to be here today and to get an opportunity to speak to you, and hopefully beyond the sandwich and chips, which, by the way, I would do just about anything for sandwich and chips. Hopefully we'll be able to give you all back some information about what we're doing here at the center and give you a chance to ask questions of us throughout this event. Really, again, as Terry said, there's no way any of the advances in the field, which I'll try to show some of them, could occur without people like yourselves really opening yourselves up to participate in the work that we do here. It is truly a partnership, and it would not in any way be able to be accomplished if there weren't, again, incredibly giving people, as all of you are here. It's also just great to see so many of you and so many friends and people I've gotten to know over the years here as well. So I'm going to talk a little bit about where we are now and maybe some of the near future places we'll go with treatments for conditions like Alzheimer's disease, as well as how we diagnose the disease. And I think the two really overlap with each other. I think some of the advancements in the treatments of the disease have been driven by the fact that we can detect the disease biologically in a much better way now than we could 5 or 10 years ago. So I think it's always good to start off to remind you who we are. I know you know who we are, but on some level it can get a little confusing because there's lots of centers and things that interdigitate. So the Penn Memory Center is our clinical center. It provides clinical care to patients. It engages in clinical research, and it's involved in actually a lot of educational opportunities, educational disseminating information to all of you, and educational actually for trainees, for fellows, for students, graduate students, undergraduate students, social work fellows or interns, really trying to develop the next generation of people who are going to help take care of this disease and hopefully continue to expand the people that will help take care of this disease. The Alzheimer's Disease Research Center is an NIH-funded center, of which Penn is one of, I think now 33 or 34 centers across the country, where we're supported by the National Institutes of Health to really serve as a nidus of research here at Penn, but also as part of national and international programs. And we work closely with the Penn Memory Center and actually share leadership between the centers in terms of bringing people into the research programs that the Alzheimer's Disease Research Center does. And so the Memory Center, co-directors are myself and Jason. This is just my love of putting pictures of myself up on the screen. There'll be more of me, I have lots of them. Jason, unfortunately, as was said earlier, sadly can't be here today, and I know he really enjoys this event quite a bit, as we all do. Terry wasn't kidding. This is really something that I think everyone looks forward to at the center over the years. The center, the Penn Memory Center, the Alzheimer's Center is not a small operation. As you have seen, there are lots and lots of coordinators and staff here in the Memory Center. And then we have a number of neurologists, we have several geriatricians, we have people in geriatric psychiatry, neuropsychology, and social work. So it's a very big team of clinicians. I'm very proud of the fact that if I put this slide up maybe 10 years ago, there'd be like 5 or 6 names on it, and now there's about 20 names, and this probably is missing some people. So this just shows the growth of our center, but also the importance of this condition and having clinicians and caregivers and researchers who are involved in it as well. And just to give you some scope of what the Alzheimer's Disease Research Center is, that's me again. I'm in another picture here. So I direct the Alzheimer's Disease Research Center. It's really a web of different cores within the center that are involved in all aspects of Alzheimer's disease research. So there's the clinical core, which is very tightly linked to the Memory Center, which I co-lead with Dawn Mechanic-Hamilton. But then there's also a genomics core to look at the genetics of Alzheimer's, a neuropath core to look at the pathology of the disease, a biomarker core to look at things we measure in spinal fluid and blood, a neuroimaging core to look at what we look in the brain. All of these different cores, a data core to try to pull all this data together, all of these different sort of multidisciplinary approaches to understanding the disease are required for us to kind of move forward. And again, people who participate in our studies here provide all of that data for us to use. And actually all of that data actually goes to a central place, or at least a large portion of it, called the National Alzheimer's Coordinating Center, so that we can build up more and more harmonized information across the country and, again, develop better therapies, treatments, other treatments and diagnostics. So there have been a lot of new events since I spoke last year to talk about. One is we had really remarkably, and I think we should all step back because it's been 20 years since this has happened, we had the first full FDA approval of a drug that modifies Alzheimer's disease biology. That's an outstanding achievement. And again, all of this couldn't happen without you here. A second drug had a very successful phase 3 trial, which largely, and phase 3 is kind of the determinative trial for whether or not a drug gets approved, had a very successful study as well. That data was presented around the time of the approval of the first drug, and we expect this drug to be approved hopefully in the next 3 to 6 months. It's a drug called donanemab, which we'll talk a little bit about in a minute. We infused our first patient with lecanemab, which was the first drug that was approved in November. And I think we've now treated about 65 people with lecanemab who have started infusions. We have a wait list, or people are going through parts of the evaluation. Around 200 more are as well, and I think that will continue to expand. One of the things in talking to my colleagues at other universities and other practices is it's really required a very complicated stream of multidisciplinary individuals to get involved to set up the infrastructure for these drugs, because they involve infusion centers, they involve imaging that has to be timed to the infusions, genetic testing in some, in everyone here, and other kinds of testing that goes on throughout the course of the treatment. And so it's been a very complicated process, but I think we're now at a stage where that's starting to really ramp up for us as well. And so that's moving along quite well. I just stole my next line. As of now, again, I think there's over 60 people that we've infused, and I expect that to kind of exponentially go up over time. So what about these drugs? I think that's something a lot of people want to hear about. We've talked about them before, and I can give a very brief summary. The first drug that was approved is lecanemab, or now called Leqembi, and it was shown to be effective in a phase 3 study. Again, that's the sort of definitive trial to determine whether a drug has efficacy, whether it improves a clinical outcome. That study was quite large, actually. It enrolled 1,800 people. That's a big study. And over 18 months, they followed people. And all these patients had either mild cognitive impairment or very, very mild dementia due to Alzheimer's disease. Mild cognitive impairment is when people have some memory problems, but not enough to really impact their day-to-day function. Mild dementia is when you have a little bit more memory and thinking issues, and it affects your ability to do tasks, sometimes like doing the checkbook or other more complicated tasks. You need a little bit more help. But these are very, very mild patients, people who are still functioning, fairly well in their day-to-day activities. They all had to have a positive amyloid scan to get into the study, meaning that they had evidence of one of the core pathologies of Alzheimer's disease, which are amyloid plaques. Why is that important? Well, if you look back, and I often talk about this, if you look at studies from 5 years ago, about 25% of the people in those studies who were treated with drugs for Alzheimer's disease didn't really have Alzheimer's disease. Because clinically, we can't diagnose the disease with that level of certainty without these additional kinds of tests. And so one of the reasons why people always say, why did drug trials fail for many years? One of the reasons is because you already had a quarter of your population not even having the disease that you were trying to treat. People had some other progressive condition, not Alzheimer's or vascular disease or something else, but they didn't have Alzheimer's disease. In this study, everyone had Alzheimer's disease. And what this drug did is it's an antibody, a monoclonal antibody. So instead of your body produces antibodies when it attacks things like viruses and bacteria. In this case, we give people an antibody to the amyloid that's in the brain. These drugs target different parts of amyloid, and amyloid starts off as single molecules and then kind of clumps together. This particular drug targets amyloid when it's still in a kind of fluid phase before it forms the plaques, but when there's some of it kind of clumped together. And what the drug did remarkably is this figure right here shows amyloid levels at the beginning of the study on a PET scan. So the y-axis means the higher up you are, the more amyloid you have in your brain. And people not treated with the drug already had very high levels of amyloid, and it increased a little bit over time. Although with Alzheimer's, once you start to become symptomatic, amyloid levels kind of flatten, they level out. And what the drug did was remarkably, just amazingly actually, lower the levels of amyloid in the brain to the point where some people at the end of the trial, if you did an amyloid PET scan, we would say they don't have amyloid in their brain anymore. Their scan would look normal based on PET imaging. And again, I just want to step back. That's a remarkable achievement, that one, we can actually see the pathology of Alzheimer's disease in the brain, and two, you can give a drug and remove that pathology from the brain. I mean, it's an incredible achievement, and it was a very ambitious thing to move forward into trials. And again, I think shows that we're getting to where we're really attacking the biology of the disease. All that being said, one wants to see a clinical outcome, not just what we see on a scan. On the right, though, are some things that gave us encouragement, which is that when you attack amyloid, our expectation is that you really want to slow down that protein called tau that we talk about. So Alzheimer's are the plaques, the amyloid plaques, and the tau tangles. And it's the tangles, the tau, that is associated with the symptoms of the disease. And that's what progresses in the brain and injures the brain, or at least is associated with the injury to the brain. And so I think encouragingly, we saw with a plasma and a spinal fluid measure of tau that we can also detect that tau levels were lowered with the drug relative to without treatment, where actually they went up over time. And so we're getting now some evidence not just about attacking the target of the disease or of the drug, which is amyloid, but that it's having downstream effects on other parts of the biology. And that was seen with some other measures, so tau PET, another way of measuring tau in the brain where there's tangles in the brain. There were also evidence of lowering in the treated group relative to the untreated group. And then finally, they looked at a variety of other markers of other parts of the biology, such as measures of synapses. So synapses are the things that neurons, the sort of place that neurons talk to each other so that they can allow you to think and remember things. And another thing they saw was a measure of integrity of those synapses, which go down in Alzheimer's, that they actually, this shows this particular measure, when you have injury to the synapses, the blood level of it goes up, and they found actually the blood level going down with the drug. So now we're seeing other aspects of the biology, even downstream of tau, that seem to be affected by these drugs. And so that's really kind of compelling as well. But again, really what you want to see is clinical effects. You want to show that people are doing better than they would do if they weren't being treated with the drug. And in fact, that was seen as well. So these are measures of change on a more kind of functional measure, and a measure of both function and thinking, and a measure of pure kind of activities of daily living, your ability to do things like the checkbook. And what was seen is that treatment with the drug slowed down the decline in those changes over time. Now, it slowed it down modestly, about anywhere from 25 to 40%, depending on the measure that you see. And so in an 18-month trial, that kind of makes you 6 months slower than where you would have been if you weren't treated with the drug. So it wasn't a home run in terms of therapy, in terms of stopping the disease, but just slowing down the disease is a remarkable, again, achievement, but there's still work to do. It means that these drugs need to get better, and I'll talk a little bit about that in a minute. There was this second drug called donanemab, which also completed this phase 3 trial, which I mentioned earlier. The trial was called Trailblazer-ALZ 2, and there could be some people in this room who were treated with the drug at the Penn Memory Center as part of this trial. It's another antibody. It actually directly attacks the plaques themselves, not that kind of small fluid form of amyloid. Similarly sized trial, around 1,700 people in the trial, all again with mild cognitive impairment or mild dementia. For those of you who know the mini mental status examination, that's a score of 20 to 28 on the mini mental. Age range was somewhat restricted, 60 to 85. And again, amyloid had to be confirmed with the PET scan, but they did something different in this trial, which I think may become more and more relevant in the future. They also did a tau PET scan in everyone. And tau, as I said, is the thing that drives the injury in the brain, and in some ways stages where you are in the disease, because the more injury you have in the brain, the further along you are, the less tau you have, the less injury you have to the brain. Okay. And so actually what they did was, they said, you know, we're going to see if you have amyloid in the brain, we're going to do a tau PET scan, and if you have no tau in the brain, well, then maybe Alzheimer's isn't yet really driving your symptoms too much, and you're not likely to change in the course of an 18-month trial for us to be able to see if we're going to slow down the disease. So those people didn't get treated in the trial. They had people who had high levels of tau in the brain, and they included them in the trial, but there was some concern based on earlier data that people who already have a lot of tau in their brain, that these drugs might be less effective, but they kind of categorized those individuals. And the targeted group was the Goldilocks group, if you will, of people who had the just right amount of tau in your brain. I don't know if anyone wants to, if there's any just right amount of tau anyone wants in their brain, but that's the group that really the study was most targeted off of, or towards. And this is what they saw. So when they looked at that group who had low tau or medium tau, they showed a little bit better efficacy than lecanemab, although I would take that with a grain of salt because I think it's more about who they targeted than the drugs themselves. But again, they showed slowing in the progression of the disease on clinical measures. They actually showed even more remarkable clearance of amyloid in the brain than lecanemab did, in that by the end of the trial, about 80% of people had no more amyloid in their brain. But it showed really, again, sort of as a replication almost of the lecanemab trial, that this approach slows down the course of Alzheimer's disease. Now, one of the things, this is the same slide I just showed you, when they looked at the higher tau group, there was less benefit in those people, which is somewhat what they predicted. This is actually combining the high group with the group that showed an effect, and so it may even be, they didn't present the data this way, but it may even be less in those high tau people that benefited from the drug if you just looked at them alone. So already we're starting to get some evidence of maybe there's a group to target with these drugs. And in fact, and I think this is pretty compelling, is if you looked at people who had mild cognitive impairment amongst the people in this trial. So now the more, not only people in the lower tau group, but the people who had more mild symptoms, there was an even greater benefit of the drug, to where there was almost a 60% decline or benefit of the drug over time, and actually very little decline in the 18 months of the study in the treated group relative to the untreated group. And that was seen on multiple measures. And so the benefit of the drug seems to increase with this drug the more mild you are and the lower levels that your tau are. Lecanemab, actually, they went back and did the same thing with lecanemab. This wasn't part of their trial, but they had tau PET in many of the individuals. And again, they showed very similar findings, really remarkable findings. In fact, with some measures, people treated that at the very early stages with low levels of tau didn't decline or declined very little over the 18 months of the study. And so both drugs seem to show this effect. They seem to work as you move along the disease, but the effectiveness of the drug seems to be earlier, the earlier you get. And I will also say there are already trials in people who don't have symptoms. We're involved in them. I think Dr. Vaishnavi will mention that, because if these drugs seem to work as you keep moving earlier and earlier, there's reason to believe they'll work even better before people have any symptoms at all of the disease, and maybe can help prevent people from getting the disease, or at least slow it down enough that they get it much later in life, and perhaps pass away from something else before they develop symptoms of the disease. There's still, though, a lot to learn about these drugs, and I'll talk about some of the ways we're trying to learn about this. But what happens after 18 months with these drugs? These trials are 18 months. You can't really do a clinical trial for much longer than 18 months, because these trials, to get a drug from development to completing trials, is on the order of billions of dollars for companies. And so you can't really do more than an 18-month trial. All of these drugs have open label phases where we'll get some information about what happens after 18 months, but I think we'll learn a lot more in our clinics about what these drugs do. If the treated and untreated group continue to separate over time, then these drugs are more than just a modest benefit. Then they start becoming a really, really big benefit. If things sort of stay where they are after 18 months, then I think they are a much more modest drug. And I don't think we know the answer yet to that. And hopefully we'll learn in the next couple of years. How long should one remain on treatment with these drugs? We don't know that as well. So in the lecanemab study, people were just treated 18 months through that 18 months. And then there was, again, an open label phase. There was a bit of a delay because they were waiting for the results of the study, but then people were treated continuously on it. In the donanemab trial, this is the second trial, when you cleared amyloid, you got an amyloid PET and it was negative, they stopped the drug. And so most people stopped the drug within that 18 months of the study. That is obviously very compelling and something I think we would all love, because that means that people are on treatment for a year or less than a year and a half, and then don't need to continue to take the drug. Maybe at some point you rescan them and there is some accumulation over time, and then maybe you treat again. It becomes a much more practical thing moving forward if that's the case, as well as the drug was given on a monthly basis as opposed to every 2 weeks, which is what Leqembi or lecanemab is given. The problem is we don't know yet whether stopping the drug is right. That's just how the trial was designed. And so I think we're going to learn a lot about that, because they have an open label phase. And again, we'll learn about this in practice. And I think these are some of the issues, by the way. There's been delays in approval of the FDA approval of donanemab. I think part of it has been because of the complicated nature of their study design and thinking about how they want the label to be for this drug. Again, would maintenance dosing make sense? Maybe we treat for 18 months, even with lecanemab, and then you give it monthly or every 2 months or every 3 months. We don't know yet. What are the best determinants of who's at risk? Because these drugs have some risk. For a serious neurologic event that's not reversible, it's about 1 or 2%, so it's not zero. Most of the time it is reversible, or people are asymptomatic, but they can get that edema or swelling in the brain and these little microhemorrhages that can occur. But there's a non-zero risk of a bad outcome with these drugs. Again, many families with this disease, which is devastating to a number of, to not just the patient, but to families, are willing to take that risk. That risk-benefit balance is in favor for many for the drug. But we'd like to know who's at the most risk and who's at the least risk and most likely to benefit, because ultimately we want to pick the best patients that are going to respond to these drugs. Another big question that has come up, especially as people are thinking like, well, I'd kind of like to do donanemab because it's every month, but lecanemab's here, is can you switch drugs? And we don't have an answer to that. No one's really switched these drugs yet, and we don't know if that imposes additional risks or maybe even additional benefits. In oncology, it's not uncommon to have multiple drugs targeting different parts of the cancer. Maybe since these target different parts of amyloid, maybe they would work even better if they were together, but we don't know that, and all of that needs to be seen. So we have a lot of work to do, and I will talk a little bit about how we're trying to get to some of that work. Before I do that, I'm going to shift gears, so that's therapeutics. Another big shift in our field is blood-based biomarkers. If you talk to any of us 5 years ago, the idea that we can measure the proteins that build up in Alzheimer's disease in the brain and detect that in blood, it was not even, it seemed like a non-starter to us, because the concentration in blood is very, very low of the proteins that build up in Alzheimer's, like amyloid. And that's why we've gotten spinal fluid in the past, because in spinal fluid, you're getting much more direct measurement of those proteins that build up in the brain. Well, technology has really improved for detecting smaller and smaller quantities of different proteins and molecules in the blood. And that's led us to where now we actually have blood-based biomarkers. Most of them are in research, although there are a couple clinically available. I really think in the next year or two, we will be using these drugs clinically in practice in a variety of ways, from screening and maybe even for deciding whether people take one of these drugs in the future, which would be a much easier pathway, not just at centers like our own, but actually to sort of democratize treatment across the country, because not everyone has a PET scanner, not everyone has the capacity to do the spinal fluid testing that we do. And I just want to show on the right, this is data from our own cohort, and this is using one of the plasma measures, and actually probably not the best plasma measure, in patients who had autopsy at Penn, who had evidence of Alzheimer's disease versus people who didn't. And this drug, and this is something we call a receiver operator curve. You don't have to know that, other than to know that more this kind of runs along in this shape, the better it is at picking up a problem. And in this case, it had about a 92% accuracy relative to autopsy for a blood test. And again, this isn't one of the best measures. It was one of the earlier measures, but it just shows you, again, the power of these methods. We've done work, and so this is work done in part of the Alzheimer's Disease Research Center and in collaboration with the Frontotemporal Dementia Center at Penn, to really say, these blood tests are often tested between patients who are normal and patients with Alzheimer's disease. Well, the other thing we need to know is how do they perform in mixed pathology, people who have frontotemporal dementia, Lewy body disease, other conditions, to really know whether these measures are sensitive. And so we've gathered together data from our center, from our research cohort. And if you look at cognitively normal individuals, we can measure things like astrocyte activity, which is a measure of inflammation in the brain. We can measure something called NFL, which is a measure of injury to the brain. not the National Football League, although many of them have injury to the brain, bad joke. And we can also measure this p-tau measure. And the bottom line is when we combine these measures, we actually can, even in normal individuals using these blood tests, we can start to pick up who's more likely to have Alzheimer's disease preclinically without symptoms yet versus those that don't. So the red group are normal individuals, people who are walking around and cognitively intact, where we're seeing signal on our blood tests to say they have the brain changes suggestive of Alzheimer's disease. And then we see that we can actually, this is looking at lots of patients in our center, and I think this just shows these little balls here represent an individual person, just how many people of all of you participate in research here, where we can start to get real power to get an idea about how these drugs work. And again, we were able to show with a little over 90% ability to discriminate Alzheimer's disease-related cognitive problems from non-Alzheimer's disease-related cognitive problems. So more blood-based, so now we have an even better blood-based biomarker that we're studying in our cohort. And so this is some early work by Chris Brown, who's a postdoc and a fellow in our center, where he's looked at a different marker called p-tau 217. Don't worry about the name, but we're getting remarkably good results with this particular blood test, where we're now getting up to 97% accuracy with amyloid PET imaging. And one of the cool things, and you don't have to worry about the details here, I'll worry about the details. We can differentiate, there's an amyloid-based measure where we can differentiate individuals who don't have amyloid in their brain from individuals who do have amyloid in their brain. But then these tau measures, these blood-based tau measures, also can differentiate whether or not you have low levels or no tau in the brain versus whether you have higher tau in the brain. Why is that important? Well, I just spent 5 minutes telling you about how levels of tau may influence who's going to respond to these drugs. And so if we have a blood-based test that can help us with that, that would be a really helpful tool for us to use to pick the best patients possible. All right, so here's the complicated thing. And this is the thing that our center, I think, emphasizes. It's the main theme of the Alzheimer's Disease Research Center at Penn, which is that Alzheimer's is a heterogeneous condition. Okay, it's very variable. I think any of you in this room know this. I don't know how many people say, when my mom had it, she got worse for, over 10 or 15 years. And then when my husband has it, he got worse much more quickly, or vice versa. It's a very variable disease. And this is actually data of every single person in the donanemab trial, that trial for the drug that showed those good effects. And this just shows, it's called a spaghetti plot, kind of makes you a little hungry, where it shows the line of each individual over time on these different tests. And it's just a cloud here, because different people start off a little bit differently, and they also change differently. There are some people here who rapidly decline. There are others that are much slower in terms of their decline. And so one of the real kind of key things we need to understand is who is more likely to decline, who isn't more likely to decline, and how that interacts with whether these drugs are beneficial. And one of the most complicated reasons why there's this variability is that, and I've talked about this before, is that Alzheimer's disease often is associated with other pathologies in the brain. And so this is work from our autopsy cohort here at Penn, which, by the way, just to take a step back, people who donate their brains here it's not only an incredibly altruistic and selfless act to donate a brain to our center for research, but it really, really impacts the work we do, because it allows us to link all the information we have from life to what was actually in the brain in individuals. And so these are people who had confirmed Alzheimer's disease at pathology, but this gray group were the only people who had just Alzheimer's disease in their brain. This green group are people who had two pathologies, one called TDP43, don't worry about the name, or alpha-synuclein, who you might know better as being related to Lewy bodies in the brain. And basically, only about a third of people had pure Alzheimer's disease. If you add in vascular disease, then in blue here are people who were clinically diagnosed from another center with Alzheimer's disease. And again, this non-blue group represents misdiagnosis, because I talked before that we're not great clinically in diagnosing the disease. But if you look at the blue and look at people who had something in addition to Alzheimer's, this is the sliver of people who just had Alzheimer's disease. So the most uncommon form of Alzheimer's disease is Alzheimer's disease. It's almost always associated with other pathologies. And so this is something we're working very hard to try to better characterize. And we do that here through really trying to characterize patients and people that we see, and also cognitively normal individuals, because as we know, these diseases begin before people have symptoms. We've spent a lot of time trying to classify individuals so we can learn as much as we can about them to better understand what are the sources of this heterogeneity. And so we get information in people who participate in all of our research studies here, in their genomics, whether they have other comorbidities like diabetes or hypertension. We try to learn about things like social and structural determinants of health, so things like stress, racism, issues with food capacity or ability to get food. So we look at all of those things to try to better understand what are the things that influence outcomes. Pollution is another thing we've been spending a lot of time looking at. We measure proteins and pathology in the brain with our autopsies, and we also link that to a variety of different tools that we now have. And again, if I presented this 10 years ago, it wouldn't look like this. We now have amyloid PET, we have tau PET, we have special MRI measures that can detect structure, function, vascular disease. We measure cognition with paper and pencil tests, but now more digital measures that Dr. Mechanic-Hamilton will talk about in a bit. We measure biofluids, again, in terms of blood and in spinal fluid, to link all of this back to the brain so we can better understand this heterogeneity. And we're starting to see fruits of that labor. So this is work that was done by a couple of graduate students in my laboratory, where we basically try to, we know, as I said earlier, that tau in the brain is what drives symptoms in Alzheimer's. And so we thought, well, what if we remove the effect of tau from the brain? What if we basically, and this is done through statistics, we say, what does your brain look like if we take out the amount of variance in your brain that's driven by tau pathology? What do we get? And what we get is we get different brain patterns of brain shrinkage, or people who have actually less shrinkage than we'd expect for a given level of tau pathology. So some people who seem really vulnerable have more injury than we'd expect, and some people who have less injury than we expect. It turns out the vulnerable people, based on autopsy studies we have, based on other imaging features, tend to have other co-pathologies. So they tend to be the people with Alzheimer's who have vascular disease, who have those other proteins like TDP-43 and alpha-synuclein. And it turns out the real resilient people tend to have less of any other co-pathologies. They tend to be just resilient to co-pathologies. And likely, actually, and we've looked also at brain age, which is sort of a separate change that you can have in your brain. And actually, we can take a scan, and using machine learning, I can predict someone's age with about a 3 to 5 year accuracy. What we also see is resilient people tend to have younger looking brains to begin with. And so that gets into all sorts of things like those social and structural determinants and nutrition and health, but that seems protective. And moreover, when we look at our typical Alzheimer's disease patients, which are this black bar here, and these are people who change over time. So this is, the higher up you go, the worse your thinking is over time. If we look at the resilient people, they show almost no change with Alzheimer's disease. These are patients with Alzheimer's disease over the several years of this study. Whereas if we look at the vulnerable people, they have much more rapid rates, so this red and orange group. And so we're starting to be able to disentangle who's more likely to develop more rapid symptoms versus who isn't, and who's more likely to have these co-pathologies. Another source of heterogeneity that we're looking at here, and this is also in my lab with Chris Brown, who I mentioned earlier, who does really great work, is people have different symptoms with Alzheimer's. Some people have more language symptoms, some people have more visual spatial, some have more memory. And there's this idea from mouse models that the way the disease spreads in the brain is it spreads from cell to cell, so from different connected parts of the brain, because neurons are connected to each other. And so what we've started to do is with imaging now, we can look at what's called the connectome, which is how every individual's brain is connected from one area of the brain to another. We can look at where there's tau in the brain in individuals, and we can actually, with pretty good accuracy, if we put those things together, predict where tau is going to spread in the brain in an individual. Why is that important? Well, one, it tells us what's going to happen to people in terms of the symptoms they're going to have, but it also allows us to better potentially measure what we should be targeting and looking at whether a drug is effective in them. And so, in addition to the fact that it supports this biological mechanism of spread, which I think also sets itself up for targets, so targets that are between synapses of neurons that maybe we can sort of target tau there and prevent that spread. And so I think this has, potentially really important impact. One of the things we're doing, and this is in collaboration actually with Dr. Brown, who I just mentioned, Dr. McKenna-Hamilton, and others, is something to start to study this heterogeneity in our patients. And we've gotten a gift and a grant to support this work, which is our anti-amyloid therapy monitoring study, which is also called ATM. It's not the banking thing, but it's a way for us to really start to study the patients that we're giving these drugs to in a much more detailed way, kind of like how we do with our research cohorts. And the goal is really to learn who is most likely to benefit from and or have side effects of these drugs. And I think the only way we're going to learn this is to do this in the real world, not in just a research cohort, but to really study our patients who come in with all sorts of symptoms, other things going on in their lives. We need to really measure those, what's going on in those patients. And that really is the idea of getting towards the buzzword that I'm sure you hear sometimes called precision medicine, which is to best learn in any individual who's likely to respond to or have side effects from a drug and to tailor our treatments to that. I also think by participating in this, we'll learn a lot more about ARIA, which is that side effect of these drugs, that swelling and the bleeding that occurs with them. Better about like the mechanism, as well as more information about how to prevent that and how to reduce that. So what we're going to do is any patient who's being evaluated for lecanemab or ultimately donanemab, we're going to ask them if they want to be enrolled in this study. We've tried to make it so that they don't feel like they're doing anything extra with the study, because we know these drugs are very time consuming. And so all we're doing is you get a lot of MRI scanning as part of clinically to follow people. So we're just adding additional research sequences to people's MRI scan, which means that your MRI goes from about a 20-minute MRI to about a 40-minute MRI scan. So it's just a longer MRI. When you come in for your IVs or if you give blood, that's a point where we can take blood, and so we're banking blood and running some of those plasma measures that I talked about before, including other kinds of measures as well. And so we'll have lots and lots of biofluid data in individuals. Again, a patient wouldn't even necessarily feel anything different from that. We just take some of the blood when the IV goes in. We'll also measure genetics. We're going to do digital cognitive testing, which Dr. McKenna-Hamilton will talk about. You're in an infusion center. You're getting your infusion for 45 minutes. You're bored. She has these fun games to play that actually you can test cognition with them. And we're basically going to take all of this data and apply it to our patient population and really try to better learn, again, who's going to do best with these drugs and who might not do as well, so that we can better treat patients. And I think our goal is to do this here, and our goal is actually to expand this to work with other academic centers to really get as much data as we can. And my feeling is this will enhance how we use these drugs. I think we will better find the people who will best respond to them and have the least risk. I also think we're not done yet. These drugs are the first drugs, and I think the drugs are going to get better, and I think they're going to get better quicker, or pretty quickly. So I put this in. This is data from an early phase study of a newer antibody to amyloid that gets across the blood-brain barrier using what's called a molecular shuttle. Don't worry about that. But the bottom line is the way these antibodies get into the brain, lecanemab and donanemab, is they sort of soak the blood vessels. And the blood vessels have amyloid in it, and that's what probably causes this inflammation, this swelling that occurs in the brain. This drug gets in a different way, and what was remarkable is at the high dose of this drug, amyloid was cleared in 3 months in all of the individuals. There was no amyloid after just 3 months of treatment with this drug. And there was no ARIA with this drug as well. Small numbers, there were only 10 people on this high dose, but I can assure you these drugs are going to get better over time in terms of their safety profile, maybe in terms of their efficacy, in terms of how we target, which patients we target, but they will get better in terms of their safety. There's no doubt in my mind about that. I just want to also say that we're doing lots of work to develop therapeutics here at Penn as part of the Alzheimer's Center, part of this other institute I work in called the Institute on Aging. We're doing studies and supporting laboratories here at Penn that support RNA-targeted treatments to reduce the production of protein. So this is actually a really neat study that's now in a mouse model where they're taking RNA, which is part of what how proteins are built in the brain, and targeting the proteins that are, or the RNA that produces amyloid and tau in the brain, and kind of blocking that production so that one reduces amyloid and tau in the brain. There's a gene therapy that's being looked at right now at Penn, right now doing safety studies to see if you can give a particular protein or a better version of a protein for clearing amyloid and tau from the brain. My very close colleague, Eddie Lee, has another protein that already has targets that are based on other drugs that have been that are out there for other purposes, that targets a protein that is called a disaggregase, which is a protein in your cells that actually pulls apart proteins that get clumped together. And that might also be something that would lower levels of amyloid and tau and all the proteins in the brain. I mentioned our ATM study. I will mention this one study because I think it's very cool. ApoE4 is the big risk gene for Alzheimer's, and it seems to be related to how microglia, which are the immune cells in the brain, work. And they're doing, this lab, Michael Haney, who we just recently recruited to Penn, is doing work where they can genetically engineer stem cells, and those stem cells can have an ApoE form that's more protective, and they're replacing microglia in the brain with these stem cell produced, genetically engineered microglia, so that you now have microglia that have the good form of ApoE as opposed to the bad form. And so this is in mice, it's still a number of years away, but I actually think these therapies are starting to happen in cancer, cell replacement, and mRNA obviously was developed at Penn for the vaccine. And so we are really moving into a new, more molecular era of the disease. And just to end, this is the slide of all the drugs that are in different trials right now. It's unbelievable. So there's been more of a commitment from the federal government in Alzheimer's disease, and that's actually starting to really reflect in terms of the number of trials that are in various phases of study, targeting not just amyloid and tau, but even other mechanisms like immune mechanisms and a variety of other approaches. So there's a really amazing portfolio of research. And so I am going to end there. I thank you all for your attention. You'll have plenty of time to ask questions. And I think we're going to move to our next speaker, which is my pleasure to introduce Dr. Mechanic Hamilton, who I work very closely with in leading the clinical core, and she leads our neuropsychology program at the Penn Memory Center. So thank you all.
see everyone out there. I'm joined by Morgann Adams, who is going to be doing part of this presentation with me today. And it's really a pleasure to be here, a pleasure to see all of you. And just to echo Dave Wolk, I just wanted to thank you for all of the time, the effort, the engagement that you put into this partnership in doing this kind of research with us. It is, we often talk about team science in academia, but team science really includes this whole team, and it is really exciting and really a pleasure to talk to the rest of the team today. So thank you for being here. So today, I'm going to be talking a little bit about I'm going to be talking about the ABCD squared study, which we often refer to as the D squared study, so that's how I'll refer to it today. And many of you know about the D squared study, but I wanted to just sort of start out with kind of what the motivation was behind doing this study. And this is a study that is funded by the state of Pennsylvania. And the reason why we started it and why we were interested in doing it is that we know that Black adults, African Americans, are more likely than other groups to have Alzheimer's disease and be at risk for Alzheimer's disease and related disorders. We also know that this group is underrepresented in research. And so all of the things that, we're talking about, the development of drugs, understanding all of the different aspects of brain health and disease, we need to know how that works in a diverse group of individuals in order to be able to say that that applies to everyone. And so this study really focuses on increasing representation at Penn within the Alzheimer's Disease Research Center and better understanding of healthy and disordered aging. So we want to really improve our understanding of both the risk factors and the resilience factors, the protective factors, the things that can help stave off brain changes as we age in a diverse group of individuals. It also has an aim of training the next generation of folks who are going to be doing this kind of work, so supports a summer program for undergraduates and graduates from diverse backgrounds to better understand and introduce individuals to this kind of work, to know what it is, what's needed, and to get their input, to increase the representation within our workforce and help with reducing healthcare disparities. So far, through the very hard work of the engagement team for D squared, we have enrolled and partnered with 115 participants with the goal of 160 participants. So still working hard to make those changes and to increase those numbers. And really excited about all of the work that has gone in and all that partnership that's happened so far. This is the kind of vital data that is being collected, and Dr. Wolk just talked a little bit about the data that's collected in the overall cohort, and it's collected in all of our cohorts, and this is just talking a little bit about within the D squared study. So talked about some of the standard and more novel MRI imaging measures that we have, amyloid and tau PET scans. Some of you may have already done some of these. Some of the standard and more novel cognitive measures that I'll talk a little bit more about. As a neuropsychologist, I'm really interested in measuring cognition in ways that's accurate for all people, and to be able to do that in a way that's a little bit less frustrating and time-consuming than hours of paper and pencil testing. We're also collecting a lot of information on social determinants of health right now, which I'll talk a little bit more in detail about. And also on risk factors and presence of cerebrovascular disease, thinking of the heterogeneity of the diseases that really affect our cognition, and really needing to know what all of those components are. And then, of course, genetic and ancestry information. So all of this information together is collected in our overall cohort and our D squared cohort to better understand health and brain disease and aging. So I want to talk to you a little bit more in detail about, if you'll indulge me for a moment, about our cognitive tests. So many of you here have done our cognitive tests where you come in and we ask you to remember things and draw them, and you may or may not like that experience. It can be a little bit frustrating. I see some heads shaking, not liking them. And we understand that. We know we need to get that data because we need to be able to see over time what happens with our thinking. And we need to be able to measure that reliably. And you're your best comparison, right? So if we can compare you to you over time, that is really helpful and a powerful comparison for us to be able to make. But we want to do better by that. So we've developed these, as Dave said, these fun games. Hopefully they're fun. For those of you who have taken part in the MCAP study, which is part of D-Squared and the overall ABC study, we have these 3 games on a phone that we have people do in the office, and we send them home with some people, and some people are willing to take it home and play it at home over time. And there we can really collect a lot of data quickly, 10 minutes on a phone, and that can be a really powerful amount of data and gives us the ability to really look at individuals over time. It also can be helpful in doing things like remote monitoring. So you don't always have to be coming into the office. You may be coming into the office for certain things, but maybe we can do more remote monitoring so that it reduces the burden on patients and caregivers, and that can be something that would improve all of the kinds of work that we do to be able to do more reliable remote work. The task on the right here, this is a virtual kitchen challenge. Some of you have already probably participated in this. This is a touch screen version of an activity that you may do on a daily basis, which is make a breakfast or make a lunch. It's often hard to kind of mimic real life activities, but these get a little bit closer to that and allow us to really see how someone walks through an activity like that. That's multi-step. That takes a lot of kind of organization and planning, and that information can be really helpful to identify early changes in cognition. So these are some of the kinds of tasks that we are doing with folks that you may have already done or you may be asked to do, and we thank you for doing those. So Dave Wolk mentioned social and structural determinants of health, and I just wanted to talk a little bit more about that because this is a really important component that really got off the ground with the D-Squared study and has now been implemented within our overall cohort as well. And the idea really is to collect information about all of your experiences across your lifespan, because we know that there are experiences and exposures from birth, childhood, and throughout our life that can affect brain development and brain aging. And so we want to better understand that in a diverse group of research participants and partners in this. And so you can see some of these domains, things like social supports, education, quality of education, occupation, social identity, your environment that you're in and the resources that are available to people in their environments as they're living and growing and throughout their lives. So these are the kinds of information that we are collecting. But the big questions that we're really asking are, what supports did you have throughout your life? And what stressors were present throughout your life that may have conferred some risk for cognitive impairment later on, or risk for Alzheimer's disease, or protection from those processes. So we think many of you have completed these surveys, 212 already. We're continuing to collect them. And one thing I wanted to mention is that we've already put out a paper with Dr. Shana Stites, who's one of our researchers here, on how and why to collect this information so that other groups across the country can be doing this as well and incorporating that into their research programs. And we've just recently put together some of the feedback that we got from folks on completing these surveys here, And what we've seen, with very few exceptions, is that people are willing to answer these questions, even questions that sometimes are psychologically heavy and difficult to answer. With very few exceptions, people are answering them and willing to do so. And we know that's a big ask, and really thank you for trusting us with that information and filling those out. And so we're going to continue to be collecting that. I wanted to give you just a little bit of kind of feedback on the kinds of questions that we ask and the range of responses. And so here you can see a question from early development, from childhood. How much did your parent or caregiver or adult read to you in childhood? We know that early exposures to education and different resources can affect development. We want to know how that affects changes later on in life. And on the right, you see really more a current question about stressors. So what is your current level of stress? Have you been experiencing that recently? And we know that those kinds of experiences can also affect our cognition, our brain health. And so those are the kinds of things we're trying to understand. And then just in our last couple of slides, just want to mention some of the sort of resilience factors and other things that we're asking about, which is, how much connection do you have with other people? So on the left, how many friends do you hear from, do you talk to? Those kinds of connections can be really quite meaningful and helpful in maintaining brain health. And on the right, volunteerism. And we see that in those who responded to these surveys so far, almost half of you are volunteering. And that's a great gift that people give to their community, but also something that can be really helpful and protective for the individual in terms of maintaining their social and cognitive and physical activities. So in my last slide, before handing it over to Morgan, I just wanted to show you kind of the impact that the D-squared study has had overall on our cohort. And so our cohort, this is the cohort of individuals with unimpaired cognition. It has grown quite a bit over the last 4 years. And what we have seen because of the inclusion of the D-squared study is that we've grown those numbers of Black and African American participants by about 20% within our overall cohort. And so we thank you for all of your participation. And with that, I want to hand it over to Morgann Adams to talk about engagement.
Thank you, Dawn, and thank you all again for being here today. You're going to hear that, I think, from every one of us, just lots of thank yous, because we wouldn't be able to be here and do all these things without you. That being said, Dawn set up the perfect transition, and I'm going to talk to you a little bit about outreach. My name is Morgann Adams, and I am one of the outreach coordinators here at the Penn Memory Center. And so outreach has made a lot of this possible. These numbers here that you see here. We realized that we wanted to ensure that our research cohorts were representative of the community of Philadelphia, as well as the U.S. at large. And so I'm going to talk to you about how we do outreach and what some of the numbers and things have looked like over the past year. Right here, we have our outreach strategy. So for many of you, some of you may have seen me for the first time out in the community. This isn't the first time you've seen me. Some of you might not be so familiar with my face. And so what we do as an outreach team is we actually go directly out into the community to provide brain health education. And so when we're looking at making sure that our research cohort is representative of our entire population, we're going directly into these spaces where we'll find some of these underrepresented groups. So we work with various community partners to engage with you all, the Philadelphia community at large, to promote these healthy brain habits in research. So the individuals who have met me out in the community, you may have seen me at a healthy brain aging presentation where we connect with older adults to talk about what healthy brain aging looks like, what are some signs of abnormal aging, and why is clinical research so vital and important to our communities. From there, we promote our brain health research registry and can enroll community members into a PNC research study, whether that is ABC, D squared, diverse BCID, HEAD, all the different studies, and that will allow you then to participate in clinical research. So we're over here on step 5. And then the final step in our outreach strategy is re-engagement with community members. We want to know who you are connected with. We want to engage within your communities, and so by bringing you into the PMC research programs, we're able to make other connections throughout our community. We really couldn't do what we do in terms of research, in terms of outreach, without the help of each of you. So, what does that look like over the past year? Our research registry, which houses individuals who are interested in participating in any sort of clinical research opportunity here at the Penn Memory Center, we've had 293 people join our research registry over the past year, which is huge. And that was done over the course of 33 different outreach events, whether that was participating in a health fair, whether your organization within your community was hosting an event and just wanted us to host a table. It might have been a healthy brain aging presentation where we actually talked more in depth about what healthy brain aging looks like in research participation. And with that, we collaborated with more than 19 community and faith-based organizations. And so we know that we're not the only person or only organization within our communities. There are so many great organizations and faith-based institutions that are doing really impactful work with older adults and with individuals. So we want to collaborate with those people, work with them to provide further knowledge and education on brain health and clinical research opportunities. And we've also had the growth and expansion of our outreach team, which was really, really exciting. For the past couple of years, it had been, we'd had one outreach coordinator, myself, I think shortly before that was Sharnita, and we've expanded to an outreach team of 3 individuals. So there are now 3 outreach coordinators, which just expands the reach that we're able to have, which is really, really great. That being said, I want to highlight some of the community collaborations we've had. And this list is not an exhaustive list by any means. Over the past year, we've had some really great programs and opportunities with the Alzheimer's Association, various Divine Nine sororities and fraternities, Enon Tabernacle Baptist Church, Grace Baptist Church of Germantown, lots of our state representatives, Joanna McClinton and Eamon Brown, have been really impactful, as well as some collaborations with the YMCA, United Unitarian House, and PACE. Many of you may be members of the PACE organization, which is a Pennsylvania prescription service, where we've actually been able to partner with them to send out mailing letters to individuals that might be eligible for our studies. So it allows people to learn about the Penn Memory Center, some of the resources we offer, and then reach out to us if they're interested in participating in clinical research. Again, trying to find ways and avenues that we can go out into the community and reach people directly instead of waiting or expecting individuals to come to us. And then just a brief introduction of our outreach team. So there are now 3 outreach coordinators. Myself, my primary focus is recruitment of Black and African American individuals. And we have Leah Barnes, who recently joined us. Leah Barnes' focus is really with clinical trial recruitment. A lot of those treatment-based studies that Sanjeev will go over, Davis touched on earlier as well. Leah's going to be focusing a lot of her recruitment efforts there. And then we've been super excited to expand our outreach team with Andrew Beltran as well. Andrew is working on the expansion of our Hispanic and Latino outreach and recruitment efforts. Don showed the figure earlier that showed the growth that we've been able to have amongst Black and African Americans, which is great. But what we also see is that we are not the only disproportionately affected group by this disease. Hispanic and Latino individuals are diagnosed with higher rates of Alzheimer's disease, and so Andrew is going to be targeting some of his outreach efforts to that community here in Philadelphia as well. So, as we look forward into this next year, there's still so much work for us to do in terms of outreach, in terms of brain health education. And so, as I mentioned, one of the things that we are prioritizing is outreach into the Hispanic and Latino community. This has been really exciting, again, with the hire of Andrew. We've also expanded our neuropsychology team with a bilingual neuropsychologist as well as postdoc, which allows us to be able to administer some of these cognitive assessments in Spanish as well as English. further expanding the reach that we have. We also are going to do some targeted recruitment efforts with males, just because we see that there's a lack of representation of males from Black and African American communities, as well as Hispanic and Latino communities. And then a very exciting launch of our participant advisory board. Some of you in this room are members of our first inaugural Penn Memory Center Research Participant Advisory Board. And this is going to be a group of individuals to provide feedback to us directly on things that we can be doing better as a center in terms of outreach and recruitment. Again, because we want to make sure that we are going to the areas that need it most and providing information to you all that is beneficial and helpful. So that being said, I'm going to turn it over to Terrence for our next speaker. Thank you so much. I did want to mention, I'm so sorry, that if you are interested in collaborating in any sort of way with outreach, you might have an organization or a community group that you think that we should work with, please feel free to find me afterwards. I'd love to connect more to talk about some of the needs and ways we can collaborate.
I promise I'm not speaking again right now, but unfortunately Dr. Vaishnavi wasn't feeling well and we wanted you to go home with information without a fever, and so he pre-recorded our next segment. I'll also say that every meeting that Morgan and I have before she leaves for one of these events, she rolls her eyes as I say, and don't forget, try to have someone take your picture, and I'm really glad that she did, because we had a lot of nice pictures from those events.
Hello everyone, I'm Sanjeev and I'm proud to present a brief update on clinical trials that we're doing here at the Penn Memory Center. It's my 10th year here at the Memory Center. I'm glad to see that our studies have continued to grow and expand, and we've made great advances in our field. Hopefully in the next 10 years we'll see a lot more progress towards getting to the end of Alzheimer's disease. I do work as a site investigator for clinical trials here at the Memory Center with a variety of industry companies as well as NIH-sponsored studies. So the first study we're going to talk about is the largest. It is the Aging Brain Cohort Study. So the ABC study is the largest study that we're doing here at the Memory Center. It is a longitudinal study where we collect, store, and share observational data to better understand age-associated brain aging and cognitive impairment, specifically Alzheimer's disease. We enroll people who are normal controls, patients with mild cognitive impairment, Alzheimer's, and other related disorders. This is sponsored by the National Institutes of Health as part of the National Alzheimer's Coordinator Center with 37 sites across the country. So recent updates to the study, we have increased the number of sub-studies and co-enrollments. As you'll see in the next few slides, many of the people that are enrolling currently in ABC are enrolled through other studies. We are targeting underrepresented groups because as a field, all of our studies really aren't clearly matching the patients with Alzheimer's disease. So we need to really target and focus to get a better representation of minorities in all of our studies. We're trying to provide participants with some results and feedback and trying to reduce the barriers for attending visits. Again, the inclusion and exclusion criteria is similar among many of the studies that you're going to see today. ABC is for people who are 65 and older, and right now we're focusing on underrepresented groups. ABC does require having a study partner, and in all visits, people do questionnaires, including the clinical dementia rating scale, neurological exam, cognitive testing, and blood draws. There are a number of optional sub-studies, which include imaging, MRI scans, structural brain scans, PET scans, looking for the abnormal proteins of amyloid and tau and others, microbiome study, optional lumbar puncture, and brain donation. And again, there are a number of co-enrollment studies we'll talk about. People continue to participate in the study until the end of life, and then people can choose to donate their brain if interested as well. So the next thing we're going to talk about is the ABCD2 study. This is the Aging Brain Cohort dedicated to diversity. The goal of this study is to enroll 160 Black participants with normal cognition to help increase diversity within our cohort. This is sponsored by the NIH and the state of Pennsylvania Department of Health. The visits include interviews, questionnaires, cognitive testing, neurological exam, and blood draws. And people do get an MRI and both amyloid and tau PET scans, as well as some optical imaging. People participate for 4 years, after which they may join into ABC. So we're looking for people who are 65 and older, who are African American, and who are normal in cognition. So MCAP is our app study. So this is the development and validation of a mobile cognitive app performance program for older adults. People in MCAP are co-enrolled in ABC, DE squared, and ABC. The idea is to develop this app as a cognitive testing platform and determine how reliable and valid is it in person and at home. People come in for one in-person visit and 2 weeks of at-home app use. Looking for people 60 to 89 who are cognitively normal and who are willing to and able to use a smartphone. In all of these slides, the contact information for our research coordinators will be here on the bottom right. Diverse VCID is the diverse vascular contributions to cognitive impairment and dementia. We know that people with Alzheimer's disease oftentimes have vascular disease changes in their brain as well, and we're trying to better understand what is the interrelationship between Alzheimer's pathology and vascular disease. So the focus of this study is again to increase diversity, especially among African American and Hispanic participants. People will get a clinical read of an MRI and the results of vascular risk factor labs. We're about halfway enrolled at this point. Again, looking for people 65 to 90 inclusive who have some cognitive complaint but are willing to commit for 3 years into the study. So Mismatch is another study in which we're looking to collect primarily imaging data from individuals with a cognitive impairment to try to better understand the relationship between the amyloid and tau proteins of Alzheimer's and other markers of neurodegeneration. So people will get amyloid, tau, PET, and an MRI. So we're looking to recruit 50 people with MCI or Alzheimer's over the next 5 years, and everybody gets enrolled in ABC. There is a long list of people waiting to be enrolled. So again, people may not end up getting into the study if we reach our recruitment goal. So LEAD study is a study of early onset Alzheimer's disease. So this is the first study we're looking at where people are less than 65. So it's people 40 to 64 who have mild cognitive impairment or dementia thought to be due to Alzheimer's disease. I'm going to give this another longitudinal study in which we collect biomarkers, including MRI, amyloid, and tau PET, and if amyloid is negative, FDG PET, and a whole bunch of other data. Again, this really was the first study to kind of expand our cohort to the below 65 age range. This currently closed for recruitment temporarily. This may reopen in summertime. A new study, which will soon be open for enrollment, is called Clarity. This is an imaging study trying to get more clarity in imaging of Alzheimer's disease. So again, another longitudinal study to understand co-pathologies for Alzheimer's and related dementias. We'll collect MRI, amyloid, tau, and FDG PET. People are 55 plus, and they're enrolled in ABC. So we'll switch gears a little bit. So Super Bowl is a study looking at plasma biomarkers of Alzheimer's disease. We know that plasma biomarkers really can help us to facilitate early and accurate diagnosis of Alzheimer's disease. And the goal of this study is to develop evidence for the clinical utility of these tests and identify what factors could limit the translation of these tests to real-world practice. So we're using a plasma test called p-tau, or phosphorylated tau 181. So we're recruiting up to 130 patients, and as a participant, basically what you're agreeing to is a blood draw for that p-tau testing, and then clinical disclosure and some online surveys. So the clinicians who order the study will have to complete some online surveys, and the idea is to see how much does this change our confidence in our diagnosis. And again, this is looking for people who have a suspicion of mild cognitive impairment or dementia. So ADNI4 is the Alzheimer's Disease Neuroimaging Initiative Part 4 study. We've been recruiting for ADNI really since the beginning. This is another NIH-sponsored study. And this is sort of a natural history, longitudinal study trying to validate biomarkers for Alzheimer's disease clinical trials. Looking for people 50 to 90 who are cognitively normal mild cognitive impairment or dementia. We collect a lot of biomarkers. We are focusing in this version of ADNI more on improving the representation for underrepresented groups, because the prior ADNIs, like all of our studies, really were lacking in the representation standpoint. AHEAD is the first treatment trial that I'm going to talk about. AHEAD is a clinical trial co-sponsored by the NIH and ESI. It's using a medication called lecanemab, and that's something that you may be familiar with. Lecanemab, as you know, is the first FDA-approved anti-amyloid therapy, which has gone into clinical practice. And the idea of AHEAD is to look to see whether lecanemab may be effective in people who are still cognitively normal. Lecanemab is currently approved for people with mild cognitive impairment or mild dementia. What we're looking for in AHEAD is to see if we go even earlier in people who are cognitively normal, but who have elevated amyloid already in their brain. Could we further slow down or prevent the development of cognitive impairment? So in this study, we're looking for people who are 55 to 80, again, who are cognitively normal. So the next treatment trial I'm going to talk about is the START study. So this is, again, an NIH industry consortium study. This is a small molecule compound called a sigma-2 receptor antagonist, which instead of trying to get rid of amyloid, actually tries to block the effect of amyloid on the synapses. So this is an 18 months double-blind, placebo-controlled, randomized control trial. This is an oral medication. So going back to the AHEAD study, lecanemab is an infusion study. So there are actually two arms of AHEAD. Either you'll be in the A3 or A4-5 arm. So you'll either get every 2 or every 4 week IV infusions for the duration of the study. In contrast, START is a pill study. So people will get either 200 milligrams, 100 milligrams, or placebo pills twice a day for the 18 months of the study. So people 50 to 85 who have mild cognitive impairment or mild Alzheimer's disease with a mini-middle of 20 to 30 are the people we're looking for. Soon we'll have another study called ATM, which is the anti-amyloid therapy monitoring study. This is really looking for people who are on anti-amyloid therapies, which is lecanemab here in the Memory Center, to try to get additional data for us to better understand the clinical use of this medicine and side effects and risks, and who hopefully get more data on who is going to improve or who may be at risk for deterioration with anti-amyloid therapies. So thank you very much. Again, we can't do these clinical trials without you participants. Again, we've made a lot of progress in the past 10 years that I've been here at the Memory Center, and I'm hoping we can make much more progress in the next decade. Thank you.
Next, I'd like to welcome up Alison Lynn. Alison is our Director of Social Work.
Good afternoon, everyone. My name is Alison. I know many of you. There's some newer faces to me as well, so I'm excited to see all of you. You've been sitting for a while, so I'm going to try and make this brief. I'm going to be talking about the social programs that we have here at the Memory Center, specifically the ones that are new, and then a brief review of our enhanced gamma programming. Can everyone hear me? I'll get closer to the mic. So this is our team. You've seen these names. I'm Alison. I'm the Director of Social Work. Leah is our Associate Director. Meg Kalafsky is our Programming Director. And then we have a new Social Work Fellow, Rachel, this year. So we have 2 brand new programs this year that have been really successful. We started them based on caregiver feedback and client feedback. The first is called the Caring Collective. It's a peer mentorship program that pairs seasoned or former caregivers with newer caregivers. It was created in response, and this is probably like a top 10 question that I get when I work with our participants, to talk to someone who's been there, either has been doing this or living this for many, many years or used to. Mentors go through a screening and a training. and then they provide about 1 hour a week of support for sessions lasting 6 weeks. And then of course we have monthly get-togethers for mentors for advice and support. This has been a really successful program. I've heard from people who are newly diagnosed or newly engaged in research that to get a little kind of glimmer of hope of what the future might look like, or even just some reassurance, will really cut down on the feelings of isolation and anxiety. So for all of these programs I mentioned, if you're interested in them, I have my cards up front where you get your parking validated at the table. You can also come talk to me, or my contact information will be at the end. The second new program we have is we started a social work fellowship last year. This is sort of our first foray into a fellowship besides the social work interns who we train every year. And Rachel, who is our social worker, is providing brief treatment psychotherapy. So it's a 2-year program we created to provide advanced specialized training to social workers who want to become psychotherapists. And the goal is to build sort of a dementia-competent workforce. So many of you, if you have had to look around for a new therapist or counselor, have probably had the experience of finding out that A, it's very hard to find someone with openings right now, even harder to find someone with openings who takes your insurance, and then even harder to find someone with openings who takes your insurance who specializes in either living with some form of cognitive impairment or caring for someone with any form of cognitive impairment. So we wanted to fill in that gap. Rachel meets with patients and caregivers for up to 8 weeks of free counseling. It's a brief treatment model. And since June of 2023, so not even a year really, she's seen 56 clients. She's currently working with 17 people, and she has 34 on the waiting list. So for reference, I've been at the Memory Center for almost 10 years now. I saw psychotherapy patients for a good chunk of that time. This is more in 1 year than I've seen in my entire career, because people tend to stick around, which is great, we want that, but this allows Rachel to see as many people as she can, and also hopefully help people who traditionally might not have access to psychotherapy. On the horizon, I'm very, very excited to announce that we, and by we I don't just mean the Penn Memory Center, although we have sort of spearheaded this proposal, but we as a Penn Medicine entity have been selected as a site for Medicare's Innovation Center's guide model. We're going to provide services starting in July 2025 is the goal. And some of the benefits for all of you who are here this morning or afternoon are an annual respite care allowance, which is provided by Medicare. So many of you are probably aware that if you're looking to bring in some outside help into the home, Medicare typically does not cover that. Medicaid is the primary payer for long-term care services. So if you are eligible for Medicaid and you have services through Medicaid, or if you're not eligible for Medicaid and were previously unable to access services, Medicare is going to give you this allowance basically every year to use for in-home help, adult day centers, any other kind of respite services you need. It's going to start with a home visit, a personalized care plan, and regular check-ins with a care navigator, so a much higher touch than my team is able to provide right now, as well as access to a 24-hour hotline. So if it's 3 o'clock in the morning and you're having a problem, you'll have someone you can call. Just so you know, eligibility, I'm sorry Jersey folks, I'm one of you, but you have to be living in Pennsylvania, and you need to be living at home with some sort of dementia diagnosis. TBD whether we're going to be able to get folks with mild cognitive impairment to be eligible. I think we might be able to sneak in. You're enrolled in Medicare with no Advantage plan though, or both Medicare and Medicaid, so you can have dual eligibility. And not enrolled in another Medicare program, such as hospice or a PACE program. Right, so that is in the works. That's about the amount of information I know about at this time. So as we start to gear this program up, we're going to be distributing a lot more on this. Everyone who is eligible within the zip codes is going to get a letter from Medicare saying, hey, call us up as soon as we're able to. So you will hear about this when we're ready. Our continued programming, which if you've been in our research studies for a while, you've probably heard of before, we do offer monthly support groups, classes for caregivers, and we have a caregiver lecture series once a month over Zoom, and we have video of all of these different topics, and that video is available on our website. So we have just a library of special topics in living with and caring for someone with cognitive impairment. We have a mindfulness and meditation class that's typically run by one of our social work interns. We have online grief yoga. Many of you I know from seeing you at our Memory Cafe, which is a once-a-month social engagement for our patients and research participants at Christchurch Neighborhood House in Old City. And we usually have some kind of music or arts programming there. And then of course Meg's program, Time Together and Weekly Smile, where she pairs our research participants and our patients with college students from Penn, Temple, Drexel, all over for companionship. This is our website. It's the Programs and Services tab on the top of the page, and you can find a full description of everything that we do on there. Okay, so what else can you access besides this group programming? I want to make sure all of you know that these one-on-one services with my staff are not just for clinical patients at the Memory Center. They're available to anyone who participates in research as well. And in the past year, is that my cue music? Okay, I thought I was doing so well. Thank you, Morgann. So I have been getting a lot more referrals from research coordinators over the past year or so, which is fantastic. That's the way to get in touch with us. So you can contact us directly, or you can say to your coordinator, hey, could you put me in touch with a social worker, and they'll make it happen. But all of these services and programs are free. We can work with you one-on-one to help provide disease education, planning for the future, coping with an emotional response to your or a loved one's diagnosis, assistance with symptom management, communication tips for managing family dynamics, for managing caregiving, for managing pretty much everything in your life, because it all comes back to this. And then of course referrals to local resources and respite care. So again, please contact your study coordinator if you'd like to be contacted with us, or you can contact me directly, although I will be out this summer. For those of you who can't see me, I will be out this summer. So please do not contact me between June 1st and September 1st. If you need assistance during the summer, please contact our associate director, Leah, and Leah will take great care of you. So thank you everyone again. Yes, please take pictures of our contact information. It's up on the website as well. And thank you so much for participating in research.